Perfect. Well, hello everyone. Good morning. Thank you for having me and letting me present a little bit about how we are improving iPSC cultures on the reagent side. So I'm going to be talking how we empower early iPSC discovery all the way to clinical applications. And I'm Evie. I'm the business development manager for stem cell and organoid research. So if you guys have any inquiries about our reagents, please feel free to reach out to me. So like I said, I'm going to be talking a little bit about our iPSC support portfolio, and then touching really focusing in on one of our applications or examples of our applications, which is in type 1 diabetes and how we're improving beta cell differentiation and a little bit of our organoid applications. So at BioTechne, we're really trying to provide solutions at every stage of the workflow for you when it comes to iPSCs. And that really encompasses from cell isolation and reprogramming through cell engineering and expansion to differentiation, characterization, as well as in vivo monitoring. So this is really an end-to-end support, ensuring that you have consistent and reproducible results. And you can see that we have hundreds of different products that can really support each step along the way. So our large portfolio of products really allows us to support probably one of the most widely varying steps of iPSC workflows, which is really the cell differentiation and characterization component of that workflow. And at BioTechne, we know it's really critical to confirm that your stem cell differentiation is robust and that it delivers cells with the right phenotype that you're trying to produce. And we really want to support your process through a reliable way, right? So we offer animal-free iPSC expansion medium in small molecules and proteins, as well as really highly specific antibodies for detailed cell characterization. And we develop all of these reagents in-house, which really allows us to pay close attention to their performance and their consistency when it comes to your cultures. So at BioTechne, we're really committed to providing the stem cell and organoid research community with innovative solutions that cater to your guys' needs. Not everyone here may be familiar with all of BioTechne's product offerings, but you're likely familiar with one of our many different product lines or brands. And so I particularly cover the RUO and a little bit of the GMP stem cell culture portfolio. So that includes R&D systems, recombinant proteins, antibodies, Talkris, which is our small molecules brand, and then as well as Accelerate iPSC media and supplements and Coltrix extracellular matrices. And then you guys have met my colleagues who are covering more of the single cell dispensing and clonal selection part. And then also our other colleagues, which are really covering the spatial biology component of our product line. So type 1 diabetes is a big hot disease right now. So it's one disease that our products are currently enabling researchers to address with new therapeutics. So type 1 diabetes is an autoimmune disorder where the immune system begins to destroy insulin producing beta cells. And one of way that we can think of really treating this disorder is by replacing or protecting these cells, right? So the schematic right here shows pancreatic islets, which house different hormone-producing cells, including alpha cells, which secrete glucagon, beta cells, which secrete insulin, and then delta cells, which secrete somatostatin. So the outcome really here of this disorder is cytotoxic T cells killing the beta cells, leading to no insulin production. And so this is a really multi-level autoimmune disorder that brings in different immune cells, and they're all working together essentially to kill beta cells and leaving the patient without any insulin production. So this is a huge clinical burden with more than 1.6 million Americans living with type 1 diabetes. And today, we're seeing an increase in the incidence of this by 2 to 3% annually. So it's one of the fastest growing chronic conditions in youth. And there's a big limitation in our current approach to the therapy of type 1 diabetes. So insulin therapy really helps manage glucose levels, but it can't really fully replicate the physiological insulin release that the beta cells do. This leaves patients really at risk of hypo or hyperglycemia and long-term complications, especially in the future when we see that insulin demand may outpace insulin supply capacity, making a huge case for us to really think about what therapeutics can solve that problem. So currently, there are kind of three lanes of thinking of how we can treat type 1 diabetes. The first being we use donor-derived beta islets to infuse into recipient patients. But now we're really creating an allotherapy that treats one disorder, but now replaces a whole other plethora with other medical anomalies, right? So we are now putting in donor cells into a recipient patient, and they're going to have to be on immunosuppressives for the rest of their life, which comes with all sorts of complications. Similarly, xenotransplants or pig beta islets, which resemble human islets very well, still carry the chance of rejection, right? So we might eliminate the issue with sourcing donors by using porcine beta islets, but we're still really maintaining that big issue of rejection. So the biggest promising field that is now rising for a therapy for type 1 diabetes is the use of patient-derived or donor somatic cells that are reprogrammed into iPSCs, differentiated into beta cells, and then transplanted into the patient. This has a huge promise to overcome donor shortages and carries really low levels of rejection risk because you are using autologous cells. But obviously, this is technically complex, it's expensive, and it still really requires refinement to generate really stable and fully functional beta cells. So during embryogenesis, pluripotent epiblast really give rise to three germ layers that we know, right? Ectoderm, mesoderm, and endoderm. The foregut endoderm folds are created after you produce all three lineages, and from that is where the beta islets begin to form from. So we really have a good understanding of the in vivo development of beta cells, and we're really trying to recapitulate that in vitro in order to create these therapeutics that are going into these patients. So BioTechne's R&D scientists really are continuously working to meet industry demands by assessing trends and then validating our products across identified workflows of interest, like producing iPSC-derived beta cells, that ensure clinical translation is obtainable for our customers. We have taken the time to establish many protocols and can help support you guys with different workflow optimization or building a protocol from the ground up using our products. But briefly here, we took iPSCs and seeded them into 24 well plates and allowed them to reach 95 to 100% confluency before inducing differentiation. Our differentiation protocol used all small molecules, proteins, media, and coating from our product lines in order to really support the terminal maturation of beta islets. So I'm going to go over a few different ways that we really improved the differentiation aspect of pancreatic beta islets from iPSCs using some of our newer product lines. So to start, N21 media supplement is a supplement that we offer, and this is added into our media. And this was found to really increase the efficiency of pancreatic cell differentiation. So here we can see that the differentiation of PDX1 positive pancreatic cells from iPSCs was elevated in the presence of N21 max. And so this is an example of where speaking to one of my colleagues or myself can really uncover some of those little supplements that we have that will really result in large improvements in your culture. Here we see by day 10 of differentiation, we're already getting about a 20%, 15% increase in the number of PDX1 positive cells. And literature shows that that N21 max was actually really able to increase monohormonal beta cells by 40% at the end of their differentiation. So what looks like a small change here actually ended up being a significant change at the end of differentiation for our customers. And this was shown in a stem cell paper. Another way that we have been really improving iPSC differentiation pipelines is using our new designer proteins. So in addition to our extensive catalog of high quality research and GMP grade proteins, we now offer a new line of recombinant proteins that are engineered using AI technology, and they're really designed for optimized stem cell cultures. Some of the ones that we offer that are less relevant to beta islets but are still important to mention are heat-stable versions of IL-2 and FGF basic, which are ideal for extended cultures or reduced feeding schedules with fewer manual touch points. And I'll be touching on the FGF basic in a second. The one that I really want to highlight here is our hyperactive version of Activin A, which really promotes efficient endoderm differentiation in human iPSC lines. And it allows you to do that using up to 4 times less protein than the wild type Activin A. And then we also have a Wnt R-spondin dual agonist surrogate, which is a well-defined 2-in-1 reagent that helps you substitute for conditioned media and or the addition of 2 individual proteins. These are just some examples of how AI is really helping to revolutionize protein engineering for improved stem cell culture. And we're excited to continue to bring innovative new products to streamline workflows, improve reproducibility, and add consistency to cell culture. So here I'm showing how our FGF basic is actually really, really stable and is allowing us to do reduced feeding. So you're putting less hands on your cells. You're not risking contamination from splits and from media exchanges because you're allowing your cells to grow in the same media for a longer period of time. And here we can see on the left-hand side that on our daily feeding schedule, our heat-stable FGF2 was able to maintain higher cell counts than wild-type FGF2. And even when we reduced the feeding, our heat-stable FGF2 was still able to maintain the same cell count as it does with daily feeding and still improved compared to our wild-type FGF2. On the right-hand side, you can see some flow cytometric analysis, which is really showing that this daily feeding has no difference, but in the reduced feeding, so let's say that you don't want to come in over the weekend and you want to feed your cells on Friday, split them and come back Monday so that you can enjoy the beach. You can really see that here you're maintaining the population of interest and in a robust manner. The one that's really specific for beta islet production is activin A. So hyperactivin A really provides a more potent, stable, and reproducible way to induce definitive endoderm differentiation. By ensuring a stronger foundation, it improves not only the efficiency of early lineage commitment, but it's also eventually leading to a higher yield and more functional beta islet cell production, which again, those early steps of differentiation really set you up to have a successful terminal maturation step. How does hyperactivin A improve definitive endoderm differentiation? Well, it induces stronger SMAD2/3 signaling, which allows your cells to be less likely to deviate from definitive endoderm differentiation. And because of that stronger signaling, it's really directing the cells into the correct lineage of interest. And so again, not only are you able to use less of the protein, you're also leading to more definitive differentiation than the wild type activin A. So I'll touch really quickly on some ways that we can characterize the differentiation of pancreatic beta cells from iPSCs using our product portfolio, which includes tools for flow cytometry, microscopy, and ELISAs. So here we offer a wide range of products for validating and characterizing your cell phenotype. We provide the antibodies, the assays, and the platforms for confirming that cells are progressing along the right path towards, for example, functional beta cells. In addition, these assays allowed us to really determine that our products are efficiently leading to definitive endoderm differentiation, as seen by flow here. And then followed by pancreatic beta cell maturation, as seen with the appearance of NKX and PDX expression, as well as CHGA expression and the appearance of aggregates by day 14 to 35. By the end of our differentiation, we have different functional assays that we provide, including flow, immunofluorescence microscopy tools, as well as ELISA tools. So here we can see basically that we really integrated our different analytical assays into different time points in order to evaluate the differentiation of our cells. So first, we use flow cytometry to really understand and quantify the different populations of interest and the ones that really mark definitive steps in our differentiation. And we can see that we are able to produce with our workflow high levels of each population of interest and very pure populations at that as well. So we also obviously have the tools for immunofluorescence microscopy in order to understand our final product. And we can see here that we're producing the correct beta cells that express C-peptide, as well as delta cells that are expressing somatostatin. And we were able to overlay those and really understand where our cells are in spatial orientation, as well as the numbers of each of the cell types. So finally, we're able to implement our simplex human insulin cartridge here in order to understand whether or not we're making functional pancreatic beta islet cells. And we're able to see that with our workflow, we created beta islet cells that are responding to glucose and secreting insulin in response to glucose. And so again, I was able to show you that we have all the tools for these analytical assays in order to understand your differentiation pipeline and to understand the phenotype of the cells that you're producing and through different ways. And so we're really highlighting that we can provide all of these tools for you to choose what method you would want to do for analyzing your cell products. So one thing I know I'm cutting it close on time, but I want to highlight that we're also able to support you through your organoid applications from 2D to 3D. So organoids are self-organizing 3D structures. They offer a more physiological relevant model than 2D culture and are now really being used and applied in areas for disease modeling or drug screening, as well as potential transplantation. So we know that the NIH is talking about organoids and moving away from animal models, and organoids are really going to be the future of therapeutic development. So BioTechne realizes that organoids and iPSCs are the future of therapeutics for a variety of diseases, and we have been working really hard to build up the necessary tools for researchers to achieve amazing advancements in the organoid field. Many of our products for organoid and iPSC differentiation workflows have GMP formats available, allowing us to really support you through every step of your workflow. So here's an example of us generating pancreatic ductal-like organoids from iPSCs using our BioTechne reagents. Researchers have achieved significantly pure progenitor populations, which then produce really nice terminally differentiated pancreatic cells with high expression of key markers as shown below. And that's by day 32. So we're really able to take our 2D cultures and move them to 3D using our products without having really large levels of optimization for each transition. And the last thing I want to highlight is really that BioTechne recognizes the importance of high quality recombinant proteins at every stage of stem cell research, whether you're performing basic development biology studies or complex disease modeling or cell therapy applications. We're proud to offer research grade, animal-free preclinical grade, and GMP grade R&D system recombinant proteins to provide you with the right combination of quality and documentation to meet your individual project needs. All of our proteins are rigorously tested for lot-to-lot consistency, bioactivity, and bioburdens to ensure quality and reproducibility. This really ensures you get the same protein performance every time, eliminating another variable from your experimental workflows. So who do you reach out to at what stage? So we're aligning with our partners across the pipeline from discovery, preclinical, IND enabling, and clinical application teams. And our R&D, our BioTechne team is really here to support you at each stage. So for myself, I cover the RUO products. So I would be here to help you for the discovery phase and the preclinical phase. And I would hand you off to our business development manager for cell and gene therapy for the Southern California territory. That's Milena Perosi. And she covers all GMP product lines and can help you with that transition from preclinical to IND and then to obviously clinical manufacturing. So with that, I just wanted to provide my email and a little bit about what we can provide you, whether it's onboarding onto our products, helping you create a new protocol using our products, or helping you find the right reagents for your unique workflow. And with that, I'll take any questions. Thank you.
Thanks, Evey.